Okay, welcome back. So we are mostly past all that math, and for now I want you to think about something different. I want you to think about turbidity, which is also known as optical density or cloudiness. And this happens whenever you have small particles that scatter light. It happens in clouds. It's what makes clouds look like clouds. It's what makes milk look like milk. It's what makes muddy water look muddy. And bacteria make water turbid. If you have enough bacteria in a test tube, for example, like the ones I was drawing for lecture 1, if you have enough bacteria, it will get cloudy. And I was kind of implying that in lecture 1, but now I kind of want to show you. So let's talk a little bit about that turbidity. So the cells scatter light. That means it bounces off of them in random directions, and some of it doesn't pass through the liquid because it bounced off the cells. So if we look at the light directly passing through, what we're going to see is the fraction of the light that directly passed through, minus whatever bounced off the bacteria. And so we can quantify that missing light using a spectrophotometer. And that's one way we estimate bacterial populations. We put a test tube in a spectrophotometer and it tells us how much light is being scattered. And if we look at a culture of bacteria sort of from the side, it will glow a little bit, and that's how we'll know there are cells in it. So I want to show you this. I have a demonstration sort of. Okay, hi. So this is a bottle I made a long time ago that has a rheoscopic fluid in it. And a rheoscopic fluid is made of little crystals that they kind of mimic what bacteria would do in water. And they're meant to make it, to make turbulence visible. So that's kind of what that does. And I only have it because it's cool. I mean, I don't need this for anything, but I keep it because it's cool. But it also mimics bacterial turbidity. And so I want to kind of show this to you because I've always had a lot of trouble helping students understand this. But let's say that there is a light source. It shines at you, it's very bright. And if this light shines at you and something passes in front of it that doesn't scatter much light, the amount of light doesn't change much. So it gets a little bit darker, but it stays pretty much the same. So at least it's supposed to. If I do it slowly, it gets a little darker. And then that's because some of the light is being scattered by this glass and by some of the dirt on this glass. Okay, so if we have something very turbid, it's going to scatter a lot of the light. And so if I shine this light at you and I put this turbid thing between you and the light, it gets a lot darker, right? That light is much brighter than this. But because this is scattering the light, that light couldn't all get to you. A lot of it gets scattered in different directions. So that's what it would look like if it could get to you. That's what it looks like minus the light that gets scattered. So in terms of light getting scattered, this is a trick we use for seeing bacteria in test tubes. The idea is if I shine the light from the side, if I shine it like this, the light isn't getting to you. If I hide it like that, it's not getting to you at all. Because it's pointing away from you, the light rays are going a different direction. But if I put something in front of it that scatters light, some of that light is going to get scattered at you. So this is going to light up, like so. And that's all light that's being scattered randomly towards you. That's what you're seeing. If I take that away, the light disappears. So we use this trick in the laboratory to figure out whether cells are growing in our media or not. We hold it up to a light indirectly and see whether it glows. And that is what I'm showing on the next slide. These are test tubes with different amounts of bacteria, and they're illuminated probably either from the side or from above. I don't know where the light is coming from, but it's definitely not directly pointed at you. And so these are glowing because part of the light is scattered towards you. So this test tube does not have any visible bacteria growing in it. So there aren't enough cells to scatter enough light that we can see with our eyes. But this one has enough cells that they scatter light that we can detect. And more cells, or higher and higher population size and cells per milliliter, makes it glow more and more until it starts to absorb the light. So this is what they would look like if we're putting them into a spectrophotometer to figure out how many cells there are. Right. So one thing you should know is that this tube, we can't see it scattering light, but that doesn't mean there aren't cells in it. That just means there aren't enough cells to scatter detectable light. And you can actually have a lot of cells in water and it will still be crystal clear. You can actually put roughly 10 to the 6th cells per milliliter in water and it will still be crystal clear, or optically clear. So how many cells is 10 to the 6th in English? Just pause and say it before I do. So 10 to the 6th is 1 million. So in every milliliter of water, we could hide 1 million bacterial cells and no one would see them. No one would see this glowing stuff. So we might ask, how many bacterial cells could hide in a crystal clear water bottle? So how big is your water bottle? Mine is a 750 milliliter bottle. So if we multiply 750 milliliters times 10 to the 6th bacterial cells per milliliter, how many cells is that? And I want you to answer that in scientific notation. And again, I'll put the answer on the last slide and I'll make a short video showing how I do all these calculations, but I want you to do it first. And if you took math a year ago, scratch your head and remember the math. And if you took math 10 years ago, see if you can figure it out. It's probably still in there somewhere. So go for it. You can do it. Okay. The last thing I want to show you in terms of turbidity is a thing that happens because of exponential growth. Because bacteria always double at the same rate, whether there are 2 cells or 2 million cells. And this is kind of showing you the same thing I showed you way back here, where you have a lot of numbers you can't read because they're too small, and then boom, it goes all the way to the top of the graph. Well, this has implications for public health. This has implications for things like food safety. Imagine, so again, no detectable bacteria, slightly detectable, boom, lots of bacteria. This is what we would see over time if it's doubling. So I put some numbers here that kind of mimic what this would be. I don't know the actual numbers for these tubes, but I can estimate. And so there's a question we can ask, which is, how many cells do we need before we start caring that they are there? Is 1 million total cells enough to make a person sick? Or do you need 1 trillion? That's a big difference. And that number, like how many do you need to make a person sick, really does vary over extreme orders of magnitude. There's some strains of cholera that will only get you sick if you eat literally 10 billion cells. And others, there are some E. coli strains that can make you sick if you eat 10 cells. Most are somewhere in between. And so bacteria that cause things like food poisoning, if we think about it, we normally keep food in conditions where they can't grow. We normally keep food either hot when we're cooking it, and so it kills them, or cold, so it stops them from growing. But if we keep it somehow at temperatures where they could grow, if we say, take some gravy out of the refrigerator and put it on the counter and leave it there for a few hours, a small population of bacteria can start to double. And it goes from being small to being small times 2 to being small times 4. And it stays small for a long time. And hopefully, by this point, you cook it or throw it away. But if you don't, if you leave it at a temperature where bacteria can grow, eventually they'll reach a point where they can start making toxins that would give a person food poisoning. And that just has to do with how this exponential growth works. And so this is what we see in the laboratory. If we're trying to grow these cells to a certain population size, it's hard because it goes from nothing we can see to more than we wanted very quickly. But it's the same thing you should imagine happening in your food. If you have something like gravy or soup and you leave it at room temperature for a long time, you go from these undetectable bacterial populations that can't hurt you at all, and we eat them every day, to a population that is no good at all. So, sterilize your food before it gets to this point. All right, so that's fun. So what happens if we actually do incubate cells, bacterial cells, in growth medium and let them grow? What happens in one of those test tubes? Well, they don't grow forever. They don't double forever. At some point, they run out of nutrients or their waste products build up and kill them. And it's a little more complicated than just always doubling at the same rate. So they will go through a series of phases where different things are happening. One of them is the logarithmic or exponential phase, where they double every doubling time. This is what we've been talking about. And this is what we think about when they keep doubling and doubling, going up exponentially. But before that happens, they have to do things like adapt to their environment. They have to make proteins, make enzymes that let them digest the nutrients around them, things like that. So it takes a while for them to adapt, and then they grow exponentially, and then something happens. Either they run out of food or their waste products start to accumulate and poison them, and they go into what we call stationary phase. And these are the two growth phases we think about, because this is what we study in the laboratory. We study cells when they're healthy and they're growing as fast as they can. That's traditionally what microbiologists study. But in nature, this is what most cells are doing. Most of them are kind of at the limit of they've already eaten all the food they can eat and they're waiting for more food. And then after a certain amount of time, they start running out of even the ability to just maintain themselves, and they start to die. So we can graph this, and you'll see sort of how long these phases take typically. I'm going to show you a graph with logarithmic transformation. So the y-axis, again, is going to be the logarithm of the population size. So what are the numbers going to look like on the y-axis? I mean, are they going to be big numbers like 10 to the 4th or small numbers like 4? You tell me, and then come back. Whoops. So here's our graph of growth in what we call a batch culture. That just means a bottle or a tube that is limited in size. And this is the logarithm of the population size in cells per milliliter, and this is time. And so, and then this is 6, 7, 8, 9. That's 10 to the 6th, essentially, 10 to the 7th, 10 to the 8th, 10 to the 9th. So we have two different graphs in two different colors. We have this first green turbidity graph. So this is what we would see if we were measuring the amount of light that is scattered by the cells. And this red one is what we would get if we were actually counting the cells that were alive at any given time. So there's some kind of lag phase, and then the cells have made everything they need and they start to grow exponentially. That means they start to double as fast as they possibly can, and their doubling time is constant through this whole exponential phase. And what this is showing is that once the population gets to a certain level, the cells absorb too much light and the turbidity kind of maxes out and we can't measure any more turbidity. So the turbidity graph kind of ends before the viable cells graph ends. So then, the turbidity graph kind of makes it look like they went into stationary phase early, when in fact they go into stationary phase like here. They've run out of food, or their nutrient or their waste products are poisoning them, and they can't double. And then as cells start to die off, they don't scatter light. But this is weird stuff that we just don't understand very well. But the death phase is a very slow, long process. But again, death phase is exponential going down. So instead of measuring a doubling time, we could measure how long it takes to divide in half. And that would be constant. So that's growth. That's it for logarithmic growth and the math for logarithmic growth. It's good stuff to know about. We will use it again, and you'll see it in Labster, and if we were doing a face-to-face lab, you would be learning this backwards and forwards. So I will see you at the next video, which I think will be quicker and more straightforward.